Improbable Destinies, Chapter 12, The Human Environment, by Anmol Cheema and Jonghwee Park.
This chapter was mainly focused on convergent evolution. A quick summary of what we will be discussing is that studying convergent evolution can help explain environmental phenomena and provide solutions to combat the impacts they have on different species. Some topics that came up quite a bit were convergence versus contingency, the role of predictability in developing practical and medicinal solutions, and the impact of human-caused changes to the environment. The evolution component of this chapter was mainly focused on convergent evolution, along with some other subtopics we will talk about. The first experiment had to do with Pseudomonas aeruginosa, which is a bacterium that is especially lethal in cystic fibrosis patients because they have especially thick mucus, which makes it harder to eradicate. But another reason was that they also have post-colonization evolution as it adapts to its new environment. This poses the question of if they evolve in similar ways when adapting to similar environments, in this case human lungs. Danish researchers reasoned that convergent evolution of populations in the same environment could be strong evidence of adaptive evolution. This is because microbes tend to use the same types of genes to adapt to the similar environments. Therefore, they looked for genes that were repeated multiple times in different cystic fibrosis patients. They found that up to 53 genes were possible candidates for convergent adaptation, and they reasoned that relying on convergent adaptation can indeed detect genes involved in pathogenic adaptation. One topic that showed up was convergence versus contingency. It was found that the overall repeatability by which P. aeruginosa adapts to CF patients is actually relatively low, and by focusing on genes that only mutated convergently, they were overlooking adaptive changes that occurred in only some patients. In some experiments using strains of Mycobacterium tuberculosis, phylogenetic studies showed that resistance evolved convergently many times. This of course supported convergence. However, a closer look found that this is only true for a small number of strains, which was a better indication of contingency. This pattern could also be seen in microbial adaptation. In a superstrain of E. coli with 5 mutations in the beta-lactamase gene, they found that in any one strain, one mutation would eventually lead to a second, which would lead to a third, and so on until all 5 were the inevitable outcome. They showed predictability and supported convergence. However, if a different gene propped up first, this would change the entire pattern, which would eventually lead to a different outcome, and many different outcomes were possible in this route. This supported contingency instead. Other evolution experiments in this chapter brought up some other important topics, one of them being the evolutionary arms race. One example is that when E. coli are exposed to the antibiotic trimethoprim works by disabling the DFHR gene in E. coli. So in response, the E. coli modify DFHR. And another example are pesticides. Insects often evolve resistance by preventing binding to cadherin, so researchers modified Bt toxins in pesticides so that they were able to bind to other proteins which bypassed cadherin altogether. Another important topic was natural selection. Convergent evolution due to human-caused change was first identified as a response to pollution in our environment. One response to this were that moths often got darker to blend into this new polluted environment. All of these topics together pose the question, will populations convergently return to ancestral conditions if we restore the environment to its original state? It is too hard to tell, but we do know that convergent evolution can definitely play a role in figuring out what genes are needed to introduce to a certain species that is facing a certain type of problem.
The medical application of this paper revolves around the idea of convergent evolution and how it is important to medicine. Knowledge of convergent evolution will give us a better understanding on how pathogens attack humans and what medications or treatments are needed to get rid of these pathogens. Why is this the case? If harmful bacteria adapt in the same way or in other words, convergently evolve, we can study the mutations they adapted and produce a therapeutic drug. On the other hand, if the bacteria does not adapt in the same way, it becomes significantly harder to create a drug, as we don't know how the bacteria will evolve, and because of the high unpredictability. Now I will give two examples on how convergent evolution and repeatability apply to medicine. My first example is the P. aeruginosa experiment. As mentioned before, P. aeruginosa is a harmful bacteria in the lungs of humans with cystic fibrosis. If this bacteria adapted or evolved in the same way in the lungs of people with CF, this repeatability would make it easier to make therapeutic drugs and medications. However, most of the identified genes experienced mutations in less than half of the 34 patients. Thus, repeatability of this bacteria in lungs of humans with CF is low, meaning that there is little to no convergent evolution. This will make it harder to make medications as we do not know how the bacteria will evolve or adapt. My next example is the E. coli experiment, also mentioned before. For a quick recap, the E. coli were placed in grass chambers with one of three antibiotics. After genome sequencing, the researchers compared each population with its ancestral population. All five populations exposed to trimethoprim evolved in similar ways. Trimethoprim disables the DHFR gene in E. coli. Therefore, the counter strategy of E. coli is to modify DHFR. Most changes in the five populations were in DHFR, meaning that there was high repeatability and convergent evolution. This makes it easier to make treatments as we can predict in which way the E. coli will evolve. The medical application conclusion for this chapter is that some predictability is still significantly better than no predictability. Even if not all microbes adapt using the same genetic changes, the fact that some do is important information. By learning the mechanism of adaptation, we can devise medicinal countermeasures where the identified gene is involved. By using genome sequencing, we can find out whether the infecting strain has a genetic change in question. If it does, we can unleash the therapeutic hounds. If not, we can look for other possible causes. The medical application conclusion is perfectly summarized by Roy Kishoni, whose work has been essential on this topic. He says, quote, "Even a modest predictive power might improve therapeutic outcomes by informing the selection of drugs, the preference between monotherapy or combination therapy, and the temporal dosing regimen to select genotype-based treatments that are more resilient to evolution of resistance." End quote.
Now we will be talking about structures and processes that contribute to different outcomes for different groups. Convergent evolution due to human-caused change has had a great impact on the environment. One part of this is just different environments in general. For instance, strains of P. aeruginosa that we talked about earlier adapt to different environments and are therefore genetically different and acquire different mutations. Another important factor is agriculture. Microbes and pests evolve convergent mechanisms of pesticide resistance. This leads to a need for more solutions to counter this resistance, and these solutions are often very costly, but are necessary to sustain agriculture. Therefore, some countries with better financial support systems are more successful in tending to these needs and the needs of their citizens. Another important factor is pollution. For instance, the moths that we talked about earlier, and also soils that have to adapt differently to high toxic levels. Another example is Atlantic killifish, which are able to thrive in highly polluted areas. However, not all killifish or moths or soils can adapt the same, and the ones that do have a better outcome than others. Another example of structures and processes is selective pressures. We impose selective pressures on species which results in strong selection against certain traits. Trophy hunting is one example. Trophy hunters often tend to prey on the largest, most magnificent species. This has led to smaller ornaments and weapons evolving in species to combat these new conditions. Another example is the economy. We also see these selective pressures in fisheries. Catching fish is also size selective, and this has led to the average fish size decreasing. This has become a major economic problem because the number of fish in the population does not increase in order to compensate for the smaller size. This has led to lower fishing yields, and this is harmful to countries who depend largely on fisheries for both food and economy, such as China, India, Indonesia, and the United States.
Structures and processes that can be changed to support more equitable outcomes. In this case, when we talk about equitable outcomes, we will be talking about restoring a species to its natural or original state before they convergently evolved unfairly from selective pressures due to human-caused change. One solution is to restore the environment to its original or ancestral state. This works in some cases. For example, the peppered moth in many places lost their trait of speckled countenance due to air pollution caused by humans. The solution was to eliminate air pollution and restore the environment to its original state. After this, the pepper moth re-evolved their speckled countenance. Another solution is to help the species help themselves by introducing genes that will allow them to adapt to a changing world, otherwise known as gene therapy. We can know which genes to introduce to a species facing a particular environmental challenge by looking to solutions that have worked repeatedly for other species and finding the best candidates for genetic rescue of imperiled species. One specific case for an equitable outcome or restoring a species to its original state before human direct or indirect contact are the size of the fish in the sea. Because of fishing, fishes have decreased their size so they can elude being preyed upon by humans. Thus, as a solution to this very specific case, there were development of non-size selective nets, throwing back the very largest fish to maintain their genes in the population, or maintaining fishing-free zones where large fish can thrive and export their genes for large size to fished areas.
In order to promote further discussion, we have come up with a list of questions. Feel free to pause and read these questions to yourself. Thank you.